Hello everybody. In this lesson I'm going to talk about the antigen and antibody and their structure. So it is very important before we start talking about how antigens and antibody reacts each other, we must understand how their structures are, and we are going to see that which are the parts of the antigens or the antibody that involves in reaction. So now let's look at what is antigens. So far we have understood that antigens are the foreign particles. It is also called a pathogen or immunogen. So normally they cause harm to the host body. So any foreign particles that enters to the body of the host and that harms, they are called antigens. Now there are certain things that you need to keep in mind while you learn this antigen and antibody structure. Now what is very important is also here is how is the foreign particle is being identified in our body. Like for example, today you have heard about COVID-19, the PCR test. What does PCR test means? How do they conduct this PCR test? And now in order to identify that the sign and symptom of sick people is COVID infections, they need to have a certain procedure. So this procedure must be known. So how do people, how do the people identify the foreign particles that infects the host? Okay, so that is what you have to keep in mind. All right, now in order to identify the antigens or the antibody, for example, when you are trying to know what has infected your body, first thing that people try to investigate is whether your body has got antibody or not. If there are a lot of antibody in your body, it is understood that your body has got infected and your immune system is fighting to get cured. So more antibody present, more the presence of antibody in your body, that is an indication that you are sick. The another one, there are certain pathogens that do not show much of an antibody. However, there are antigens which directly indicates the presence of the pathogen itself. For example, some of the bacteria that affects our body would be directly seen in the blood. So there are testing kits which will help to identify the antibody of the particular virus, or there are testing kits that will help to identify the foreign particle itself. What cause harm to the host? Obviously it is a pathogen, but as such the whole pathogen itself is sometimes may not be harmful, but these pathogens body parts becomes infectious, or it becomes a very virulent to the body of the host, meaning some part of the cells becomes very toxic to the body. Therefore sometimes it is not necessarily the whole organisms itself, but sometimes the product of that particular organisms is at times becomes very harmful. Therefore here it is mentioned that the antigens will have coats, capsules, cell wall, flagella, fimbriae, are at times very toxic, and sometimes it produces some toxic chemical as well. So most of the time this happens with the bacteria. Okay, and also there are pathogens that cause infections like worms, but here worms we never call antigens, because antigens are those uh, pathogens or immunogens that enters to the body right inside, because here we are talking about the specific defense, not the external defense. These pathogen has already got inside your body, and our immune system is working to fight against this. So this is not pathogens like a worm. All right, this is difference you have to understand. Worm is it can be external infection, but these antigens like cold, fimbriae capsules, this must be an internal infection. Okay, now what you have to understand is these are two structure of antigen and antibody. Now there is two sites where antigens and antibody binds to each other. For example, in case of antibody or the immunoglobin has got a paratop on the variable sites. On the variable sites, later on you will understand in the mouth part of these antibody, let me call it right now. Here, these regions and these regions of the antibody has a paratop. This paratop is has such a shape that exactly complements with the shape of the antigen. So these regions of an antigen is called epitope. So this is an antigen and this is antibody. This is an antigen. So antigen has got a structure which can directly fit to the antibody structure, that is called epitope. So you must understand epitope is the structure of antigen and paratop is the reacting regions of antibody. Okay, now to understand little in depth of the structure of antibody, there is two reasons. One is constant regions which will never change for any of the antibody, almost all will have the same structure, and this is the variable regions where you will find these regions have got a different structure for different antibodies. Now there are 4 different chains, protein chain that makes the antibody. The external 2 chain, these external 2 chains are heavy chain. The internal one, this and these are a light chain. As I said to you, these regions is a variable regions, and these regions is very reactive with the antigen. So any antigen that come across, the antibody will react from these regions, and this region remains always constant. The 2 things that you note here is one, towards the variable regions, if you look at this terminal is made up of ammonium groups, ammonium groups, or the ammonia group, NH2 group is attached on this region, whereas the constant regions will have carboxylic group. Please understand that these structures helps them to react with antigen. Likewise, this is the structure just to indicate how the antibody would look like. In reality, antibody will be like this, and if you want to show, it can be like this. So these are there is a heavy chain, there is just now it is being just shown an opposite way. There is inside one is a heavy chain, outer one is light chain. Okay, and as I said, there is a site for antigens to combine with the antibody. Likewise, you can look at this, how the antigens has complementary in its structure to fit into the antibody. So this structure is an antibody, and this is an antigen. So antigen comes and fit into it. So paratop and epitope is very important here. Now one thing I forgot to share to you is here this 4 chain, heavy chain and light chain, they have a bonding in between. You just look at the sulfur disulfide bond. Here there is a disulfide bond, here there is a disulfide bond. These bonds keeps them together, right? So this is what is very important to know about how they are, these 4 chains are kept together. So this is just to indicate how they join each other. Just look at, these are the different reactive regions where you will see the 4 chains come together and strongly bind each other, right? Now what you have to understand is sometime like these regions, different regions that you need to understand is FC regions, FAV regions. FC regions is a constant regions, fragment constant regions of the antibody. Now these fragments, two heavy chains and 2 light chains, are bound together by a disulfidal bond. And FAB is this is fragment binding regions. Okay, fragment, these regions is variable regions. So in these regions the antigens binds with the antibody. Okay, there are 5 different types of antibody. You just look at these. This is IgM, then IgG. IgM is the first one, is this IgG, this is IgM, IgG. Then you look at, this is IgA, IgE and IgD. These are the different antibodies found in our body. IgM has got 1, 2, 3, 4, 5 antibodies joined together to form Ig, and these antibody would, this antibody IgG is a single one monomer of antibody, and here IgA has got a 2 antibody joined together, and IgE is 1 monomer, and IgD is 1 monomer. You'll find these has got a different fragments and has got a different binding site. If you look at here, there are 10 binding site, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Because each antibody will have a 2 binding site. Look at this, there is 2 binding site, there are 4, there are 2, there are 2. All right. Likewise, there are these antibodies found in different parts of your body. So you must read this. This is not that every antibodies are present in every parts of your body. There is an IgA is found on your tears. Likewise, you have got these IgMs are normally found when there is a lot of infections in your body. So these quantity of this particular antibody increases depending on the type of infection in your body. Right. So what you have to understand is there are 5 different types of antibodies. They bring reactions, or they fight with the pathogens depending on the type of infections. Okay, then this is just to show you how each antibody joins to form a dimer or the polymer. So you just look at, this is the single antigens which has got variable regions and the constant regions. When 2 antibodies join together forms a dimer. Okay, IgA. So dimer is IgA, and this is when there is 5 antibodies joined together and forms IgM. Okay. Now what are the different types of antibody depending on its ability to infect? So there are 3 types. Antigenic property of the immunoglobulin. Sorry, this is immunoglobulin. When we say this is antibody itself is not an antigen. At times these immunoglobulin represents the immunogen or antigens. Okay, and then based on that it is being divided into three different types: isotype, allotype, and itotype. And this also shows there are certain differences in the composition of amino acids, and also the differences in the L form and light chain and heavy chain forms. Okay, so next is just to recall some of the questions that I have noted down here. Please go through this question, like what is antigens, how does antigen binds with the antibody, how many different types of antibodies are present, why antibody is tested for any reaction or the infection, how is HIV tested, positive or negative, what is PCR test, why do PCR test is conducted for COVID-19. Okay, and there are many more here. Even it is being asked to differentiate between the paratope and epitope, isotype and allotype, antigens and antibody. Okay, so with this I like to end. Hope this will help you to understand the uh, different types of antibody and their structure and how it participate in the reaction. Thank you.